utility of it. So my name is Emile Chimusa. I'm associate professor at the University of Cape Town, and I have a great passion in human genetic variations in terms of drug response, treatments, variations, and disease susceptibility. So today I'm going to actually give you a bit of overview on polygenic risk score, and I'll show you some benchmarking work that we have done and some results so far we got on some infection diseases and also general population for UK Biobank. So as you know, genetic variations reflect human history actually, and we know, all of us, we know that 100,000 years back in time, human migrate from Africa and move around the world, and that's actually impact or contribute to the variation. And we all of us know different factors that actually contribute to variation that I've already say in terms of disease susceptibility, treatment and drug response. So those are actually also mutations that occur often and it's random, and natural selection, population bottleneck, admixture, and more other factors. So those factors are well done, but that led actually genetic has actually today huge diversity problem that need to be solved. Just imagine that if you have no information about Michael Jackson, no news at all, you never heard about him, and actually right now this gentleman appear in his room, and I have to ask you if Michael Jackson is African or European. So based on what we know about race by just the skin color, the type of the hair, you can use that to actually predict that, say, look, this gentleman is European, So that may tend to be wrong actually in reality, So now, if you got the DNA of Michael Jackson, now you use a kind of machine learning tools, you can be able today, we use that to predict actually the ancestry. And then you can tend actually to say he's an African American, where some part of his genome come from European ancestry and some part of his genome from African ancestry. So therefore, diversity is a critical for equitable translation of genetic studies. So we come from long path just to understand this kind of variations. So a lot of omics data initiative has been taking place since 1970. That was the era of DNA sequencing technology, starting by Sanger sequencing and then move forward. And I just highlight here for you just recently, since 2009, there's quite a lot of and rapidly advanced in generating data. A lot of consortium has been taking place, and many initiatives, UK Biobank, 100,000 genome from UK, 100,000 genome from Australia, from China, and also African genome variation project that's come to play. So all of this data has been generated. The question is still how many data we need actually to capture variations, and to understand this complexity of biological complexity, and also how many sophisticated machine learning we need to actually leverage this data that's already there to understand variation. Those are actually different questions that is still unsolved and is still actually a kind of a dilemma in this era. So a part of data initiative, there's actually a lot of approach has been generated, starting from 1911, that was the beginning of genetic linkage studies, and then move forward to 2005, beginning of GWAS era, 2008, microbiome study started, and till today, and the question is what next today? What can be next approach to that can actually allow us to understand the variations, and that variation actually can have impact or application in a clinical setting, So so far the progress has been done so far, but however, most of the study has been conducted in European context. As you can see from this picture, take this picture from my recent publications that show 81% of genome-wide association studies has been conducted in European. You can see in the red over there. And only 3% in blue here in Africa, that's genome-wide association has been conducted. That is really show a huge disparity in the genomic studies, and that gap actually need to be filled. So in terms of data generation and in terms of appropriate method that can account for diversity of different populations. As we know, genome-wide association is studies based on the concept of common diseases and common variant. And it's also leveraging the linkage disequilibrium. And it's well known that the population undergone population bottleneck has a long range of haplotype or linkage disequilibrium than a population who are diverse. That is, I'm talking about European one, they gone a population bottleneck, and they have a long range of haplotype, and potentially GWAS has been generated and can be run properly to this kind of populations. So while African population have a high diversity and high admixture level, and that will require actually an appropriate GWAS approach to capture this kind of genetic characteristic. And in addition, African population are well known to actually undergo also selection pressure that can also some things to actually to capture in different methods of disease scoring statistics. So far, the question is still, even though the GWAS has been done in European context, European populations, but why there was a lot of method or tool of GWAS being actually implemented, as you can see on my right panel over here, you know, since 2007 until today, last year. So there's more tool has been actually developed. So to actually capture the so-called stratification issue in GWAS and other potential issue that can eradicate the false positive and also boost the power of GWAS. So, but the question also still remain on those different tools. What can be the appropriate tool to be applied to diverse populations like African populations? So each of those tools has been benchmarked independently, but there was actually no way or no study actually benchmark those two in African populations. So we take a step forward to do so, whereby we actually simulated case control and with, of course, causal SNPs that the data case control that mimic African as a structural populations, and also case control that actually mimic European populations, and also admixed population, where we simulate five-way highly admixed individuals in five-way fashions. So what we did actually, we also tried to see the rate of false positive in most of public tool, common use GWAS tool, as you can see on x-axis of those plot over here. So the first plot diagram here show three scenario. That's mean on different sample size: 500 case control, 1,500 case, and 5,000 1,500 control, and also the reds line here is the plot of 2,500 case and 2,500 control. So those are actually different color. So on y-axis, we try actually to plot how those tools actually can generate false positive. So that we have a baseline on the simulated causal SNPs, and we run GWAS on those different data based on different samples and on those each tool, and we actually compute the proportion of false positive. As you can see, even for African samples, those different to Emax, Gmax, GCTA, Blink and so on, all those tools have always a certain proportion of false positive in African population, in European population, and also, of course, also in admixed populations. So then we see our observations across different populations, even including admixed simulated populations, that Emacs that actually implement a kind of mixed model within a kinship matrix that's actually try to account for relatedness between individual within your a cohort has a low rate of false positive. In addition, we try to understand how the replicability of GWAS conducted in European population, in African populations. And we run meta-analysis using fixed effect model here, and also random effects, random models, and also binary effect model in European GWAS, simulated GWAS, and African GWAS. So our observation, we have seen that if the effect exists in both two populations, European and African populations, the appropriate approach to use should be fixed effects and has a low rate of false positive. The reason why to assess false positive is because there's a great potential of utilizing the result of GWAS in further downstream analysis in kind of like polygeny risk score, where there's a quite expected application in a clinical and a diagnosis and a deep phenotyping and also clinical trial that I'm going to actually talk shortly about. So there's a persistent challenge, although there's a many tools that's been actually developed, and I'll mention most of two of them that people tend to neglect. Is the first one is inefficiency of GWAS in distinguish between inflation from the common non-bias critical relatedness to the true signal of polygenicity. So when we do QC in GWAS, we tend to do as much as possible to reduce population stratification. By doing so, we are losing a lot of data that may actually explain polygenicity. The second one is the imperfection of asymptotic bias distribution of a mixed model or a logistic regression that actually impose us to remove SNPs with low malloc frequencies. We are losing a lot of those rare variants that can actually explain polygenicity, or can contribute to the risk predictions. And even though today there's excitement to use whole genome sequence to cover more region of the genome, yet when you conduct GWAS, we always remove rare variants. That is the challenge that still remain, and still remain how to resolve this asymptotic bias in mixed models. And also the question is still how far so far current GWAS model can account for different type of interaction, like a host microbe interaction, gene regulation, and host genetic interaction, that's actually known to actually contribute to the host phenotype. So now, nevertheless, even those challenges still persist, actually, there is a way to leverage GWAS efforts into routine clinical use, and also we can use all those GWAS efforts to actually enable preventative or predictive medicine so far. And the common method is known to be polygeny risk score, which is actually the estimate of an individual genetic risk or reliability to a threat or a diseases, and that's been computed according to the genotype profile of an individual and also relevant causal SNPs from GWAS. So these formulas are actually where you have the effect of the causal SNPs and the individual genotype, and you try to aggregate all those small effects, and then you can actually estimate the risk or the genetic reliability of individual, and of course compare it against the rest of the individual within the populations. So today, PRS has taken a lot of attention from media, clinical applications, from the research, and also from the funder perspective. So it's actually important considerations, which is actually we have to bear in mind that causal variant effects are mostly shared across populations. And the way we are aggregating or sum up the small effects of individual within its genotype, the question is how you model these weights here, the beta, and this actually comes from GWAS summary statistics, and a different method has been generated to actually model this way people can account the differences in Mallow frequencies, the linkage disequilibrium, the model also family history, environment, and other factors. And that's how I'll describe in the next slide how multiple tool has been generated just to the way to model this kind of sum or this kind of weight within the genotype. So there's quite, as I was saying so far, this PRS have actually potential to into the application at the routine clinical use and to enable preventive and predictive medicine, and also with, into the application of differential diagnosis screening, and also for deep phenotyping. Someone want to actually do phenotyping, you can use PRS actually to do to conduct a deep phenotyping, and also for clinical trial where you want actually to have a participant with high risk. You can actually apply polygenic risk within a bunch of individuals, estimate polygenic risk, and rank individual with a high potential risk based on particular diseases. So that is how PRS can be used. So far, there's, as I say, based on this weight, actually trigger a lot of research to generate more tools that can try to model Mallow frequencies on how many SNPs, causal SNPs need to be summed up to actually determine the risk of an individual. Those are different questions, and those different questions have triggered also different tool development. So far, there's quite a lot of tools so far has been developed since 1990 until last year. There's still more tool of polygenic risk score that's been developed to capture the way to actually model the weight. But also one of the things is those tools has been developed and been applied in European cohorts, as GWAS has been conducted extensively in European descent populations. So now here we try to benchmark those tools here in African populations known of high diversity. So we actually simulate case control at the first instant just to see how accurate those tools can produce a predictive power, a comparative predictive power in African populations. So we simulate case control, and also we simulate a true proportion of variant explained, which is actually over here, and run each PRS tool on that summary statistic that we simulated, that actually coming from case control African data. And we can see over here, most of those tools have a similar magnitude of predictive power or variant proportion of variant explained, and all of them, they are far away to reach the true simulated predicted power or proportion of variant explained. So that's means there's something is missing in current tool to actually improve the PRS predictive power in African populations. Here at this instant, we actually use African trend data and African target data summary statistics, and none of those two actually reach, a true predictive power. So we was thinking maybe there's some genetic characteristic that is missing in this tool, and we try to actually to evaluate the relationship or the impact of selection on effect size and on also allele frequencies, and to see if we capture selection on its impact on effect size and on also allele frequency, if that model can increase the predictive power. So this is actually what we got over here in this plot B. Under strong selections to a high trait value, one can actually expect a more U-shape, as we see over there, a U-shaped side frequency spectrum, and a high trait increase also allele frequency. However, the reverse is true as well. Under strong selection, there are a few causal variants since more have fixed, but a high fraction of novel rare variants are trait increasing, which result in the mean trait increase allele frequency being lower than 0.5 down on the plot that is there. So therefore, the message out of this simulation, where we try to understand the relationship of selection pressure and effect size and the spectrum of allele frequency, is that strong selection results in a weaker effect, and that can limit deviation from the optimal trait value, which may help to actually explain polygenicity. So from here, actually, we have actually been convinced that if we can model, when we are weighting the sum in a polygyny risk score, if we capture selection, the impact of selection in effect size. So in this, we can see over here that the predictive power of polygyny risk score can actually increase, particularly in African populations. So in doing so, we try also to figure out if someone use a European cohort as a training models, and African population or other diverse population as a test model, how the accuracy, the behavior of PRS can be. So here we go. We try actually to model PRS by modeling additive effects of global ancestry and also local ancestry and the SNP effects, and also their interactions like SNP effects also with ancestry effects. That's how we try to model. Once we model that, we actually apply this model by implementing a less penalized regression, where we compare PRS predictive accuracy between European ancestry from UK Biobank as a trained models, and compare that to East Asians in a test as a test model, where we use kind of 8,636 sample from South Asians, 1,800 East Asians, and 6,983 African across 26 phenotypes. So this plot over here, as I was saying, European on x-axis, that was actually used. We used European as in training models, and South Asia as a test model. We can see that the correlations, the predictive power, the predictive accuracy is quite improve when you are using a trained cohort closely or genetically related to your cohort in the test models. And when we come to the African, we see there's a huge dispersion, across all the 26 phenotypes over here. So the take-home message over here is that the genetic risk predictions accuracy decay with increasing the genetic diversity. The more genetic diversity is between the train and the train or discovery cohort and the target data. We actually go further on to actually try to understand the prediction accuracy across multiple cohort that is in the UK Biobank, so the continental ancestry. And as I was saying here, you can see the predictive power decay within the genetic diversity. The more the population is diverse, the more the predict accuracy of PRS decrease. So we look on a UK Biobank on only African ancestry, and we see that there is actually this kind of decay or differentiations of predictive power across within African populations, and is actually looks for us, it tends to us to be arbitrary, but also there's bias that's actually are not actually meaningful here. And we actually also see that the natural human evolution or the selection pressure can actually explain these differences of predictive accuracy among African population as there is here. So now, having all this information, we now use a cohort infection diseases, or HIV, in the Botswana populations. As you know from that the HIV have a high prevalence in Southern African population, as you can see over this worldwide map in red here, high prevalence of HIV. And for us to actually perform progeny risk score in these populations, we did actually use the whole genome sequences of 338 samples of HIV positive and HIV negative, type 1, and of which we have 226 HIV positive and 112 HIV negative, as you can see from this PCA plot case control over here. We perform a population genetics to understand the genetic variations in the Botswana population by including other African populations. So clearly our whole genome sequence of Botswana show that Botswana is actually sitting in the convex of quiescence and is West Bantu African populations, which actually makes sense for population genetic perspective. And we conduct GWIS on that. We found some SNPs to show up at level of genome-wide significant. And all of those SNPs actually, they've been nourished in the blood, and also they are located in intergenetic regions of the genome, and also precisely they are in mtDNA and nDNA regions. So we take these summary statistics to actually perform progeny risk score, where we actually use our method where we actually account for ancestry, in additive model, the effect of global ancestry, the effect of local ancestry, and the effect of SNPs, causal SNPs, and also their interaction of ancestry and the effect of ancestry interaction with SNPs interactions, and also the model that we account for selections. So surprisingly, we got some results that show actually a very tight correlations between false positive rates and true positive rates. Bit of correlation that's seen that there's no actually the accuracy is actually promising here. And we compute the relative risk where we found 48% of the samples have a high pollution risk score. And in absolute risk, we actually found 20.7% of the samples, regardless of HIV positive or HIV negative, to have high risk score. So this is actually some things that show our potential by according ancestry in Africa and selections in the method of pollution risk score. So the take-home message here is that PRS pollution risk score differ across African population, and that is arbitrary. But these biases are not meaningful. And the second thing is that the neutral human evolution or selection pressure can explain some of the differences in pollution risk score. So at this point, there is actually a way forward where pollution risk score can actually enable the predictive medicines, but this is still a question on how actually can be real, can be feasible, you know. So our plan here is actually to leverage or to quantify the risk prediction predictable to a combination of host genetics, host microbiome, you know, non-genetic element that is like a family history, you know, even lifestyle, that's mean nutrition, and other environmental factors, and also includes EHR, electronic health records, in the model of pollution risk score to identify actually individual at a different level of risk for developing diseases. So I think by aggregating these informations, we might improve the accuracy. And also one of our observations is, even though we implement that, as we say that the PRS can differ across population, and that is arbitrary. So a way actually that to find a way to find like a consensus approach that can leverage multiple PRS tools to have a consensus risk that come from different approach, you know. And also we need to actually improve education and training of a health professional in developing new risk communication tool. Also to consider randomized trials to directly evaluate the impact of pollution risk score and the related individual health outcome. So that's it from me today, and I have to acknowledge colleagues and students and study participants